It won't happen again, but I heard it. Okay. So now I'm going to do the formal part. Good. We ready to go, Dylan? Great. Welcome, everybody, to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Carly Lipinski. She's a fifth-year graduate student in the Department of Chemistry, and she works in biochemistry. She's going to be speaking with us about illuminating protein function by imaging colorful single molecules. This is going to be cool. Carly, I get to ask you the 5 questions. The standard line is, you can answer the questions in any way that you would like. It doesn't have to be true, it just has to be believable. Carly, where were you born?
Johnson City, Tennessee.
Johnson City, Tennessee. Is that where the dam broke?
I don't know.
Oh, okay. Oh no, that was Pennsylvania.
Yes.
Johnstown, Pennsylvania. I take it all back. And where'd you go to high school?
Oh gosh, I went to high school at Benton High School in Arkansas, as well as Tavares High School in Florida.
Very good. And then where'd you go for your undergrad, and what did you study?
I went to University of Arkansas, and I studied chemistry with a focus in chemical biology.
Excellent. And then when did you arrive here?
2018.
Good. And how did you enjoy 2020?
So good.
3rd year of grad school. I can hardly wait to have another pandemic. Yeah.
Passed all my exams. So.
Well, that was good.
Yeah.
That's actually
One
pretty good
thing.
timing. Yeah. Well, I'm looking forward to your talk. Would you please join me in welcoming Carly Lipinski to Wednesday Night at the Lab?
Thank you. Thank you so much. Hi, everyone. Thank you so much for being here. I am so excited to present to you this work, Illuminating Protein Function by Imaging Colorful Single Molecules. I'm in the lab of Aaron Hoskins in the biochemistry department, but as Tom said, I'm a chemistry graduate student. And so I'm going to give you, briefly, tell you why it's important to look at molecules, single molecules, go through how we look at a single molecule, some of the molecular biology tools we have to be able to do this, as well as our microscope components for our fluorescence microscope we have in the Hoskins lab. And finally, how we can use single molecules to look at really complex protein systems in the Hoskins lab. And so first, I'd like to impart to you how important it is to look at single molecules. And so here is a single molecule, and for the purposes of this talk, we're mostly going to be talking about protein molecules, and this is a cartoon depiction of a protein. So we're just looking at a single molecule. Traditionally, in biochemistry, experiments have been done in bulk. So you'll have a bunch of molecules together in solution, and you'll be looking at the average behavior of all of those molecules together. So what information can we get just by looking at a single molecule separate from all of that averaging in ensembles? I like to draw an analogy to a marathon. So let's say you are in a helicopter and you're looking down at a marathon and you see all of these people running. And so that's the ensemble. You have a bunch of people, you can tell that they're running a marathon, and you can say things like the average runner speed was maybe 9 miles per hour, or you can say the average time to finish was 5 miles per hour, just looking at all of those runners all together, averaging their behavior. When we look at single molecules, we can say things like, we can begin to divide this population of runners into subpopulations. And so we can say, oh, maybe there's a population of people who drank water while they were running, or there's a population of people who are walking while they're doing the marathon, or population that entirely ran. And so we can look and distinguish those individual behaviors within this large population of people. We can also separate it into populations based on rates. And so maybe some runners were running at a faster speed and they finished before another group of runners. So they finished in 4 hours versus 6 hours. And we can distinguish those populations by looking at single molecules and assigning them categories. And finally, we can begin to look at the behavior of individual runners within this population. So no longer at the population or subpopulation, the behavior of individual runners. And say things like, she ran for 1 hour, walked for 15 minutes, and then ran for another hour, looking at how fast she ran, the speed that she was going at. Or we can look at position. So she's at mile 5 within the marathon. And I'd like to emphasize that looking at the individual behavior of this runner is not possible in ensemble studies. So we lose all of this information that we're gaining by looking at an individual runner and being able to sort all of those marathoners into subpopulations. So that's what it looks like for a marathon, but how does this translate to a protein? And so again, we're looking at the ensemble level, which is a bunch of molecules all together, and we're looking at the single molecule level where we're just looking at the behavior of a specific molecule. So we can say things about rates. So for averages, for ensemble measurements, we can say maybe the average enzyme produces 50 products per minute. Or maybe we can say the reaction was 99% complete after 5 hours. So again, averages, looking at the behavior of a bunch of molecules all together. For single molecule, we can look at populations of molecules. So maybe there's a population that they're producing product at a really fast rate and a population they're producing product at a slower rate. And we're able to distinguish between those two types of molecules with single molecule experiments. And we're also able to look at a single molecule, whether or not it alternates in a rate, so it's producing enzyme or producing product at a really fast rate, and then maybe it shifts to a slow rate for a while and then goes back to a fast rate. And we're able to look at and observe that behavior. And finally, we're able to look at position for individual molecules. And I'm excited to show you later in the talk some examples of this. And again, really emphasize that we can't look at these individual behaviors and separate molecules out into populations with ensemble or bulk studies, which have been traditionally done in biochemistry. So you might be thinking, well, I can't see a protein with the naked eye. How are we visualizing these molecules? And so if you look here at the scale of what we're looking at, proteins are about 10 nanometers or smaller. And to me, that's almost an incomprehensible scale. How small is that really? We just can't see it. And so we have some really neat tricks in biochemistry to be able to see these molecules in the visual spectrum. So how can we see a single protein molecule? And the answer is fluorescence. So we have fluorophores that we can attach to these proteins to be able to make them visible in the visible spectra. And so fluorescence is a property that's commonly found in nature. And so many different organisms can be seen producing fluorescence, as well as minerals and your tonic water, if you've ever looked under looked at that under UV light. And also interestingly that I found out while researching for this talk is the spots on bananas are fluorescent when you shine UV light on them. So go home and try that before you make banana bread next time. I certainly will. So fluorescence is the property of absorbing light of a short wavelength and emitting light of a longer wavelength. And I think we first need to talk about the characteristics of light before we move on into talking about how exactly fluorescence is produced. And so light falls onto electromagnetic spectrum of a lot of different kinds of radiation. And so we have our highest energy radiation, which is gamma rays, which have the shortest wavelengths, up to radio waves, which have the longest wavelengths and the least amount of energy. And visible light falls right here in the center. And each of these gives you about the size of what a wavelength would look like at that scale. And I'd also like to point out that light has this characteristic where it can behave like a wave or it can behave like a particle. And sometimes these descriptions are useful at various points in the talk, talking about it as a wave or as a photon or a particle. So I just wanted to bring your attention to the duality of light as we're moving through this talk. So I mentioned that fluorescence occurs in nature, and I'd like to go to the example of this jellyfish, the Aquaora victoria jellyfish, which is really beautiful. So here is it under visible light, but if we look at it under blue light, you can see images of this jellyfish where it's green and it has this lovely fluorescence effect. And this is due to a protein that it produces. So the protein is called green fluorescent protein, or GFP. It has this really lovely barrel-looking structure. And so, again, emphasizing the scale, it's only about 4 nanometers. But in the center of this barrel, it has this chromophore, or also called a fluorophore, where it will absorb light and produce that effect called fluorescence. Now I'd like to talk about exactly what fluorescence is and how at the molecular level does this happen. So fluorescence is a property where you absorb one color of light and emit a different color of light. And so again, we'll look at this example for green fluorescent protein, where you have a nucleus and you have an electron orbiting the nucleus here. There are two different energy levels represented by the orbitals, so the ground state and the excited state. So when light comes and is absorbed by the electron will move to the excited state. And then when the electron relaxes back down, it loses that energy and emits a wavelength of a different length or a different color. And this property can be visualized this way, and it can also be visualized with a diagram that looks like this. And so we can describe this in another way. We can imagine that you are an electron and someone has invited you to a party, okay? And so you get all excited for the party. You go to the party, you're in this excited state, all right? You dance, you get rid of some of that energy. Dancing, you expend energy, and then when you leave the party, you release all of that energy. And so the energy that you lost while you were dancing at the party is the difference between the energy that you had at the beginning and the energy that you had at the end. And if we think back to the electromagnetic spectrum, your energy depends on your wavelength. So you change the color of light that you are emitting from the type that you absorbed based on this energy that you have lost dancing at the party.
Positive or negative?
Positive or negative. So higher energy to begin with, lower energy end with. Thank you. Okay, so GFP was a really awesome molecular biology tool, and it has been further tuned to look at biological applications. And so the properties of this protein have been changed to produce different colors and to enhance the amount of light that it's able to give off through this property called fluorescence. And so we're able to take these proteins and basically add them as a tag onto the end of a protein of interest and take really beautiful images of the human body and cells. And so this is rainbow, and so each of these different colored kind of spots is a neuron within a mouse hippocampus. And so this is just really lovely application of this protein that was found within nature that we can use as a molecular biology tool to really look at the inner workings of cells and their structures. But this isn't the only way that we can give proteins a fluorescent property to be able to visualize them at the single molecule level. So the first synthetic dye for this purpose was fluorescein, and it was synthesized in 1871 by Adolf von Baer, a German chemist. And you might have encountered this chemical before. It's this reddish-orange powder, but when you dissolve it in water, it has this highlighter yellow color. And so this was a green fluorophore, and this is the earliest example of one that we had to use for biological application sets of this. And it was really quickly put to use by Albert Kuhns, who developed a technique called immunofluorescence, where you have an antibody that's targeted to a specific protein that you want to look at. And you can attach a fluorophore to that antibody, either looking at it directly with that antibody or with a secondary antibody which recognizes the first. And through immunofluorescence, you can look inside a cell and see where specific proteins are located. And so here's an image where you're looking at the cytoskeleton of cells, and you can see in green the structure of these cells. It's targeted to a specific protein in the cytoskeleton. And so this is a really powerful technique for looking at proteins within cells and has also been used for diagnostic applications, presence of proteins in disease states or not disease states. So it's been really great. And additionally, I'd like to point out that some of Albert Kuhn's images that he has taken during his work are shown directly behind him. So you can see all these different images and cell morphologies that you've been able to now image, specifically looking at a single protein. Okay, but these dyes had a problem because you're just looking at a green dye, and that happens to be an issue inside cells. Your cells contain so many different compounds, and these compounds some of them do have fluorescence properties. And so when you try to image a cell, maybe in the green spectrum, you might get something called autofluorescence. So your cells have this property innately. And some sources of autofluorescence in animal cells are collagen and elastin, and in plant cells are chlorophyll and lignin. And so here's an image of some plant cells from pine trees, and there's no fluorophore here. The fluorophore is a component that's endogenously present, or always present, in plant cells, which is called lignin, and you can see the structure here. And so if I wanted to image something with a green fluorophore that was located within the cell wall of these plant cells, I just couldn't see it because there's so much fluorescence that's present there in the cells to begin with. And so we needed new tools to be able to look at different structures within cells and avoid this property of autofluorescence. And so new dyes were synthesized in the 1980s. They gave us an expanded tool set to be able to take colorful images. These dyes needed to have certain characteristics. They needed to be non-toxic, membrane permeable, and bright and long-lasting, so we're able to have signal above cell autofluorescence background, as well as be able to image them for long periods of time. And Alan Wagner was a chemist who synthesized these dyes. And he took inspiration from photography. So in the early 1900s, film was orthochromatic, and it lacked the ability to capture wavelengths in the red spectrum. And it also had poor ability to capture in the blue spectrum. So all of your reds turned out as black on the film, and all of your blues were overexposed. And photography shifted to panchromatic, and there were dyes that were added into the photographic gel matrix that allowed the film to be able to capture light in the red wavelength and produce panchromatic film. So Alan Wagner started with these chemicals from film and was able to synthesize dyes that were bright and long-lasting and relatively non-toxic, called Psi-3 and Psi-5. And these are dyes that we continue to use in the lab today and are commonly used in our lab. So that was a great innovation from photography into using dyes for single molecule imaging. And so today we have a really expanded tool set where we can choose fluorophores we need for our application. We can choose fluorophores in the red range, in the blue range, or the green range, pretty much anything we want to be able to get the images that we need and look at the proteins that we want to. And so now that I have taken you through how we're able to visualize a single molecule, a protein, we're able to put dyes on it or fluorescent proteins, I would like to take you through our single molecule microscopes. So these are the image of the microscope that we have in the Hoskins lab, and I'll take you through some of the components. But I'd like to start with the differences between a light microscope and a fluorescence microscope. And many of you may have used a light microscope. It's something that I used in high school. And light microscopes, you are forming a dark image on a bright background. And you're doing basically the opposite for fluorescence microscopes. With a light microscope, you need to fix cells on a glass slide and stain them, and that's how you're able to visualize them. For fluorescence, you're using those fluorophores that we just spent some time talking about to be able to make these proteins visible. Light microscopes, you're looking through an eyepiece at your sample. Often with fluorescence microscopes, you're looking at images on a computer. Light microscopes have the disadvantage that you can't look at individual proteins in cells, and you can't look at living cells, and you can do both of those things with a fluorescence microscope. And finally, for a light microscope, the light source may be natural or artificial. You're using the full visible spectrum to be able to image your sample, whereas fluorescence microscopy, you want a very specific light source of a very specific wavelength to be able to excite your fluorophore of choice to produce that light. And so when we look at a fluorescence microscope, this is a very simplified version. Basically what is happening is you have a light source that you are directing onto a sample, and you are detecting that light in the opposite direction because you don't want your incident light, the light you're directing into the sample, to get into where you're detecting. And so the fluorescence is directed up into the detector, you're selecting for a specific wavelength, and then you're looking at this image on a computer. And so because I can't physically take you into the lab, I'd like to show you an image of me walking around our microscope. And you'll find that it looks very different from either of these cartoon images that I've shown of a fluorescence microscope. So here you can see, this is our table. We have all of our objectives and components mounted to the holes in this table. And you can see here there's quite a few components, and they all have to be in a specific line with each other to be able to direct the light from the lasers into our detector, which is located off screen. So as you can see, there's quite a few parts to this microscope, and it took a while to put together. Everything is custom. And I'd like to take you through just a couple of the components and what we do with them. So the first are lasers, which we're going to use to excite those fluorescent molecules, and our shutters, which we use to control the exposure to the lasers. We have a stage that's used to adjust our sample position, often at very, very fine distances, and also micromirrors that help to direct the laser light into our sample at just the amount that we want. And so we'll start with the lasers. So lasers have a specific set of properties that are distinct from visible light. So whereas visible light is composed of many wavelengths, they go all in different directions, and they diverge, so they're going everywhere. Laser light is very focused and specific. So it's monochromatic, it's a single color, it's coherent and culminated, so it's focused into infinity, and all of the wavelengths are aligned with each other. And so how exactly is this produced? And so here is a depiction of a ruby rod laser, and this is a laser that we do have on our fluorescence microscope. And within the laser, you have a lasing medium, which is the ruby rod in this case. And you have two mirrors on either side, and you have a source that is going to excite molecules within that ruby rod. And so this coil is going to put energy into the system where molecules within the ruby rod are going to go into the excited state, and they're going to emit a photon, exactly like we talked about with fluorescence. And those photons are going to be reflected back and forth between the two mirrors, the 100% mirror and the 95% reflected mirror. They're going back and forth, and very, very occasionally they will encounter a molecule that has also in the excited state, and in that case, it's going to emit two photons. And so that is the stimulated emission of radiation part of a laser, which is an acronym. And some of the laser light will escape through that 95% mirror, and that produces this wave where everything is aligned, coherent, and culminated. So you get a very precise focus, exactly like the laser pointer that I'm using. You get that very precise dot from a laser. And so one thing for safety is that lasers are separated into different classes. And so the first class are not really dangerous to eyes, and I couldn't actually find any examples of class 1 laser products, or class 1 lasers, but these are class 1 laser products. And so the lasers are hidden, and so therefore safe, and I'm sure all of you are familiar with a laser printer or maybe a CD player. And secondly are class 2 lasers. And so these are ones you just really don't want to look directly at. An example is the laser pointer that I'm using tonight. And you're protected from eye damage because of blink reflex, and also because you want to turn away from the laser. You have that innate instinct. So anything that they're using to scan barcodes at the checkout, laser pointer, those are class 2 lasers. They're not going to hurt you. Class 3 lasers are getting into the category where you can sustain eye damage, and that includes lasers used for research, such as on our microscope, as well as lasers used for like tattoo removal or other skin procedures. And finally, class 4 lasers are dangerous, and they can start fires if used improperly, so used for like LASIK treatment or micro etching. Where you're removing parts of metal using a laser. And so I just think this is kind of neat just to know what kinds of lasers you encounter on a daily basis and how dangerous they are for you. Turns out not very. But anyway, our microscope has 5 different kinds of lasers on it. So we have a 488 nanometer laser, which corresponds to the wavelength of light that that laser produces, which is blue. 532 nanometers, a green laser. 594 nanometers, an orange laser. And 633 nanometers, a red laser. And we also have an IR laser, but that's not visible to your eyes, so you won't be able to see any laser light from the IR laser on our microscopes. So I'd like to show you what our lasers look like when we turn them on our microscope. And so you can see here the different lasers going through their aligned paths, emitting light and traveling into our detector. So you can see there's a blue laser, a green laser, an orange laser, and a red laser. And it just looks really lovely. And we do all of our imaging in the dark. So I'm sitting down in the dark watching these beautiful colors while my experiment is running. And so this is what I see. What was controlling the laser light in that movie was a shutter. And this is exactly as you would expect it to be from using a camera. So the shutter is here directly in front of the laser and controls when the laser light makes it to the detector or the sample. And you can see here with the lasers on that our shutters are blocking the laser light, so for the blue, green, and red lasers. Okay, and so here's another image where we have simultaneous excitation with red and green lasers. And this is how I will do my experiments. So I'll excite for blue, and then I'll excite for red and green at the same time. And all of this is controlled automatically by the shutters. For the example of this movie, I'm turning them on and off manually, which if you'll notice, it's not quite precise. But yes, this is what it looks like when we're running an experiment with this microscope. And so another component that I'd like to talk to you about is the stage. And so the stage for our microscope is actually a local product. Mad City Labs is a company in Madison that is internationally known for their nanopositioning systems. And that basically means that you're able to very finely control the location of the slide, so you're able to bring your sample into focus to be able to image it. And so here's a cartoon of kind of what the stage looks like. As well as here you can see the top of the stage, the objective, exactly like you would picture for a light microscope. This microscope also has an objective. 3 kind of rods where we're able to fine-tune the X, Y, and Z axis manually, as well as the stage is connected up to a computer where we're able to move very, very finely in the X, Y, and Z axes. Here's the top where you can see the objective again. And finally, this is what it looks like. So it's kind of this box. It's hard to emphasize scale, but it's kind of about this big. So here is the stage part. So it's located on the opposite end of our microscope table, and you can see all the optics I previously showed back there. So this is where our stage is, and you can see this is quite a long table. It's about 8 feet long, where we're able to align the laser light from this direction over into the stage and onto our sample. Okay, so finally, I'd like to talk to you about a very specific property that we use when imaging our samples, and it's called total internal reflection fluorescence, or TIRF. And so TIRF is different from epifluorescence. These are 2 different techniques to be able to image fluorescent molecules. And so epifluorescence is where you direct a lot of laser light into your sample, and you have a very large volume where you're exciting all of the molecules within that volume. So you can see here, everything within 1 millimeter is going to be excited. So you're able to image all of those in that excitation volume. And that's in comparison with TIRF, where you're only imaging a very, very small excitation volume right above the slide surface. And this is advantageous because if you want to see just directly what's on your slide surface and not anything that's floating above it, you can use TIRF to be able to image that. And this field is called an evanescent field. And I had to look up the definition of evanescence, which is the quality of being fleeting or vanishing quickly, so impermanence. So you just have this very, very small amount of light from the lasers that's being directed into your samples. And so when we compare images for epifluorescence versus TIRF, you can see epifluorescence, you have a lot less detail. I think I actually mislabeled these. This one is TIRF, you can see very fine details. In fluorescence, you can see, you kind of like have a thicker looking cell. You can see more background within that cell. So things that are close to the surface, things that are not close to the surface. Ignore the labels above the images, look at the labels on the images. Thank you. And so a component of our microscope that makes this possible are called micromirrors, and these direct the laser light into our sample at a very critical angle required for TIRF, or total internal reflection fluorescence. And so here you can see kind of a cartoon. These micromirrors are smaller than your pinky nail. They're very, very small, and so they're directing the laser light at a specific angle up into the sample to create that effervescent field, and then back down away from the detector. And so I don't know if you guys can pick out the micromirrors in this photo, but they're right there. They're very hard to see, they're very small. And here's a close-up of them. They're these just kind of like angled, kind of look like the end of a screwdriver a little bit. And here is looking from the objective down, so you're looking from the top of the stage into the microscope. And that's what they look like. And so this kind of sets our microscope apart from a lot of other fluorescence microscopes, and is part of it being very custom. And so now that we kind of have an understanding of what fluorescence is and how this microscope works, I'd like to share with you a couple examples from other labs and from the literature, so you can get an idea of what kinds of things can we image with this technology before I talk about some of my work and work at the Hoskins lab. And so kinesin is a molecule in your cells that walks along microtubules. So microtubules are part of your cellular cytoskeleton. And so they are basically roadways that kinesin and dynein can walk along and deliver cargo to various places in your cell. And we can tag individual kinesin molecules with a fluorophore to be able to image them. And so you can ask questions like, what happens when kinesin encounters a crossroads or a dead end? It gets to the end of a microtubule, or it crosses two microtubules. And this work is done by Christine and is the first author. This was from her graduate school work, as well as the two corresponding authors on this, Alapasha and Stefan. And so I'd like to show you some movies of what this looks like, because it's kind of cool. So here is one video. This is a sped up video. As you can see, the time progressing much longer. And maybe it'll play again. Well, here's another one. And this one basically shows that the microtubules will keep going along their track when they encounter a crossroads. And the second video, with each one of these moving dots, is a single kinesin molecule walking along a microtubule. And you can see them moving and measure their individual behavior, such as how fast are they walking, and where are they positioned along the microtubule. And so as you can see, as they reach the end of the microtubule, instead of just falling off, into solution, they accumulate at the end of the microtubule. And so instead of looking at these inside cells, which you absolutely can do with single molecules, these authors have chosen to do this in a purified system. So they've purified the protein kinesin, labeled it with a fluorescent molecule, and then encouraged it to walk along these microtubules that are fixed to the slide surface. I just think this is a really beautiful video and really demonstrates how we're able to look at position using single molecule analysis, as well as rate and location. So that's one example. Yes?
I have a question about what do you mean by walking? What's actually happening there
Yes.
that allows it to move?
Yeah. So the kinesin has these two microtubule binding domains, and it will hydrolyze ATP to get it to bind to the microtubule, and it'll basically do one domain and then the other domain. So it'll bind one domain, the other domain will attach, it'll kind of flop around, and then it will hydrolyze ATP and bind again to the microtubule, and then the other domain will attach, kind of flop around, and then bind again. So because
it's walking along an ATP molecule?
Yeah, so it's using the energy from ATP to walk along the microtubule. And this is not this lab specifically, but kinesin was one of the first molecules that was really looked at using a single molecule technique, which is why I wanted to highlight it. And we have a lot of this information where we call it like walking from complementary techniques like cryo and electron microscopy, where we're able to kind of get at structure that we can't get at with single molecule, or get at in a different way than we can at single molecule. Yeah, thank you. There's one other example that I would like to talk about before I move on to what we do in the Hoskins lab, and that is looking at myofibrils, which make up the muscles in your body. And so myofibrils are composed of thin filaments, or actin, and thick filaments called myosin. And the myosin thick filaments are basically molecular motors. So we just looked at an example of a molecular motor, kinesin. We're looking at another one called myosin. And the myosin will basically hydrolyze ATP to be able to grab the actin and pull it. And that's how you have muscle contractions. And so here is a single molecule video, again, where these eye bands have been stained here in the purple, and ATP, or the molecule that provides energy, is shown here in blue. And you're able to watch in this video as individual ATP molecules come down, arrive at a myosin molecule, and are hydrolyzed and leave. And it's really quite striking. And so you're able to ask questions with this single molecule experiment, such as what happens to energy consumption in healthy versus diseased muscle fibers. And this work is done by these two authors, Matavi and Neal, both at the University of Kent. And so it's really quite striking, and I feel like I used that word to describe the last video, but to be able to localize exactly where these ATP molecules are being used within this muscle fiber, and specifically the single molecules of myosin that are utilizing those ATP, is really, really cool.
Excuse me?
Yes?
How are the ATP modified?
Yeah, so they just have a Cy3 fluorophore attached to them, actually. I think Cy3. They have a fluorophore attached to them, so we're able to visualize them.
So that doesn't affect the binding or the weight transfer?
It definitely could. So with any single molecule fluorescence experiment, when you're attaching a fluorophore or a fluorescent protein to a molecule, you have the potential to change its behavior. But the fact that they're still able to image this and hydrolyze the ATP is pretty cool. So even though it could be modified in some way, which we have to think very carefully about before doing these experiments, we can still get useful information usually. Yeah, thank you. Okay, so I'd like to move on to what I do in my thesis research. And I look at a very complex molecule called the spliceosome. And here is a depiction of the spliceosome, and different components are highlighted in the different colors. It is a space-filling model of this structure. And you may be wondering, what is the spliceosome? The spliceosome performs a very critical and special function within our cells. So when we think back to the central dogma of biology, that you have DNA that's transcribed into RNA, which contains the message for a protein, which is then translated into the protein. But before that RNA is translated into a protein, it is processed. And one of the processing steps that can take place is called splicing. And so splicing occurs on this precursor messenger RNA, which is directly a copy of what is present in a gene from DNA, to produce a messenger RNA, which is translated into a product. And when I say splicing, I would mean that these regions that eventually don't code for the protein are removed. And so you're getting removal of these regions that sometimes don't code for proteins, which is essential to producing the correct protein product. And this is really critical that this happens correctly in your cells every single time. It happens in 99% of your protein coding genes in your cells, and it can become quite complicated really quickly. So splicing patterns can vary to include various parts of the gene or exclude various parts of the gene. And so from an RNA that is being spliced, you can have inclusion of these protein coding regions 1, 3, and 5 to produce this protein, or you can have inclusion of the regions 2, 3, and 4 to produce this protein. And these proteins might have slightly different functions. They function at different rates, or they might be regulated differently, like turned off when one is turned on. And so this is very critical that this is correctly done every single time in your cells. And the spliceosome is part of the molecular machinery that is making these decisions. This process can often become dysregulated in cancer, where you're just splicing anything willy-nilly and you have all of these different protein products produced. Another cool thing about alternative splicing, which is how different protein coding regions are either included or excluded, is that it diversifies the amount of proteins that you can make from a single gene. So it's a really cool process. And the spliceosome, which accomplishes this really awesome biology, is quite complex. So in humans, it's actually composed of 153 proteins. And so all of these proteins have to come together in a defined order to be able to accomplish this process, and they have to do it every single time for every single region to be removed from the RNA. A lot of the components of the spliceosome come in packages called snRNPs, or small nuclear ribonucleoproteins, and they contain dozens of protein components as well as an RNA component. And the RNA components are essential for either recognizing those sequences to be removed or actually accomplishing the catalysis. And I mentioned that the spliceosome assembles on the regions to be removed every single time on that RNA molecule, and they assemble in a defined order every single time to produce that final mRNA product, as well as a byproduct called a lariat, which is the region that was removed. And I'd like to highlight the functions of two specific portions of the spliceosome, which are U1 and U2. And these recognize the sequences to be removed. And so we do a very specific experiment in the Hoskins lab using single molecule techniques to get at how these components of the spliceosome are interacting with their RNA that they're working on, and also with each other, to be able to get at how the protein composition changes over time, what the order is, whether or not these components arrive before or after the next components. And so we can take the RNA that the spliceosome works on and we can fix it to the slide surface and fluorescently label it. So when we look at our slide images, we're able to see the locations for the RNA. We do our experiments in whole cell extract. As you can imagine, the spliceosome is so many components, it would be difficult to do this in a purified system. So we use whole cell extract, which is basically yeast that we've lysed, and just everything inside the yeast is what we're using. Yeast have a slightly less complicated spliceosome, and yeast are very advantageous for working in the laboratory because they're very quick to grow, and they're very easy to genetically manipulate. So that's our model organism of choice. So from here, from this fixed point, this location, we're able to localize molecules of interest that are labeled with those synthetic fluorophores that we talked about earlier, Psi3 and Psi5. And then when we look at our microscope images, we're able to see locations of RNA and then localize those proteins to those locations of RNA. And we're able to watch this happen in real time. And so I'd like to show you this video where we're looking at those two components of the spliceosome that recognize the RNA portions that are to be removed. And one requires ATP to bind to the RNA, and the other one does not. And so we can either do the experiment in the presence of ATP or in the absence of ATP. And this is what you end up seeing. And so this molecule, U1, has no trouble binding that RNA with or without ATP. However, each one of these molecules, which is a single U2 molecule, only bind in the presence of ATP, and there's basically no binding in the absence of ATP. And so we're able to look at dynamics and compositional changes of the spliceosome, how these different protein components interact in this very, very complex system, and start to break that apart and learn more about the finer details of exactly how this system is functioning. And this is very much like what I do in my thesis work. And so I'd like to just leave you with how important it is to understand molecules at the single molecule level. We're able to look for rare events, and we're able to distinguish between those multiple subpopulations. If you think back to the marathon and the runners, the water drinkers, the walkers and the runners in our marathon. We're also able to look at functions of proteins in complex assemblies or environments. So the spliceosome, which is an incredibly complex machine in our cells, or kinesin walking along a microtubule. And we're also able to utilize fluorescent proteins and dyes as molecular tools to be able to bring these proteins into the visible spectrum and see them. And finally, we're able to use this to be able to detect and record the behavior of biological molecules, which is incredibly cool. There's that one. Yes?
What is the power of magnification that's involved here to see molecules like this?
Yeah, I think our microscope has about a 60x objective, which, if I remember correctly, is on a light microscope and maybe less than 100x. I don't have a great grasp of like the scale of magnification. Yeah, but maybe the scale bar is like hundreds of nanometers. It's pretty small. Yeah, thank you. Yeah, this is my lab that I work with, these really wonderful people that are so encouraging and I see every day. So Erin is my PI, and I'm currently working with students Natalie Zepps and Amory Griffin, who have been fantastic to work with on aspects of my other projects, as well as all of the graduate students who have supported me through my time in my PhD, which has been really invaluable. And I would absolutely love to take any of your questions. Thank you.
Fantastic. Let me bring the microphone to you. We'll start down here and come back.
Oh, thanks. Yeah, I was wondering, first of all, if you could say something about luminol, and then also I had a comment. Fluorescene also has some non-biological uses, primarily by hydrologists and speleologists for the purpose of tracking groundwater flow.
So you said luminol is a compound.
Right.
So when you think about fluorescence, I'm not entirely sure, but there are kind of two properties that are very similar, which is phosphorescence and fluorescence. I'm not quite sure under which category luminol falls, if it's phosphorescence or fluorescence, but a way that you can tell pretty quickly is if you shut off the light source, fluorescence disappears completely. So you turn that UV light off, it's immediately gone. I think that luminol, if you turn off the light source, it's still fluorescent. So I think that's phosphorescence, not fluorescence, which is a very, very cool molecular property as well. And as far as fluorescein, I was not aware that it was used in groundwater, so that's really cool. I know that fluorescein, one application is they use it in the optometrist office to look at your eye. So they might give you like a swab in your eye with that weird strip thing. And then be able to see things like cataracts on your eye. So that's not something that maybe I've had that done, I don't know. Maybe some of you have had that done. Kind of cool application.
We have a question on Zoom.
Oh, cool.
I see that the fluorescence intensifiers are different in the microtubule movie. How many fluorescent molecules are attached to a single kinesin molecule?
I think in that study they just attached a single fluorescence molecule. Sometimes I know in fluorescence experiments you can have uneven illumination across the field of view. So in some locations your fluorescence is more intense than the other, so in the center versus the edges. I don't know, maybe if they're walking slower, they're being imaged in that location for longer, or maybe it's multiple kinesin molecules tethered to each other and they're hauling another kinesin molecule along with them. So there's lots of different things that it could be, I'm not entirely sure. I'm happy to direct you to that paper if you would like to read more about it.
I wonder if you'd illuminate on the difficulties of calibrating your microscope, and if a train going by or a truck going by or temperature changes offers you many challenges.
Yes, so temperature changes definitely. As you can imagine, that temperature will cause the components in our microscope to expand very, very slightly. And that can cause our components to become misaligned. So you saw in those movies where you have a very straight line for the laser illumination on the microscope, some of those components can shift very, very slightly, and that causes our microscope to become out of alignment, where the laser light is no longer being very precisely moved into the camera. And so it's a huge problem. I mean, We don't often have those problems, but when it happens, it does take quite a lot of effort to realign the microscope. As far as like vibrations, the table with all the holes in it is actually very specially designed. It's on an air cushion, and so any vibrations don't actually make it to the top of the table. They're confined within the legs and the ground. And additionally, our microscope is located on the basement level to minimize any vibrations from the building. But yeah, that is a great point. Thanks.
I do want to point out to folks, there is an active railroad about 56.3 meters from where your lab is.
Yes.
And I get to watch the trains roll through from my office. And visiting kids are always astonished, like, is that a railroad? It's like, yes, it is.
Yes.
Are trains go on that thing still? It's like, yes, they do. And my major professor worked in bacteriology 50 years ago on electron microscopy, and she talked about how what Sandy just asked about was the train rolls by and you're getting videos instead of static pictures.
Yeah.
Other questions?
How do you decide which fluorescent chromophore to attach to a particular molecule?
Yeah. So the ones we like to use in lab, Sci3 and Sci5, they produce a lot of photons, so they're very easy to see on the microscope, so they're very bright, and they last a long time. One of the properties of fluorophores that I didn't really touch on is that they can photobleach. And so they can only do this so many times where you're exciting the molecule and it's producing photons. Eventually it will go into a state where it will no longer do that anymore and it's dark for the rest of the experiment. And so we want fluorophores that minimize that property so they can last as long as possible. So that's one consideration. And the second consideration is definitely color. So we want fluorophores of certain colors. Some fluorophores are just better than others. So blue fluorophores are sometimes produce less photons than maybe red or green. The same maybe goes with orange fluorophores. So it's all about like the properties. When you get into cells, it's a little bit more tricky, so you want to minimize like that autofluorescence, but we're not doing experiments in cells, so we have less of those problems. Did that answer your question? Okay. Yes.
It's a great talk.
Thank you.
So I'm wondering about data acquisition. You probably didn't have time to cover this. Data acquisition and data analysis, because you're generating huge amounts of data with these.
Yes.
And also you have to deconvolute each individual spot. So, you may not have time, but how do you go after that?
Yeah. So we're taking about a second's worth of data for every frame, and we might take up to 800 frames, and those are all compiled together into the movie. So the movie that I showed you is frames that are taken maybe about every 5 seconds. And for each of those images, we need to look and decide where the locations of those RNA molecules are, because those are our regions of interest. We care about things that bind to the RNA and things that bind to the slide surface where there's no RNA. We want to minimize, but we don't want to analyze spots where there isn't an RNA molecule. So yeah, there's software custom that can kind of like pick those spots automatically. Sometimes we also do it by hand for various reasons. And from that, you can get fluorescence intensity over time at that individual location. So you're looking at, if the molecule arrives, you'll get an increase in fluorescence intensity, and when the molecule departs, you'll get a decrease in fluorescence intensity because you're looking at that specific spot and that specific fluorophore. We have to look at all of those traces, and from those traces, we can get kinetic behavior. So like how long did the molecule stay at that particular location? How fast did it depart? How fast did it arrive? Things like that. And so we have various ways that we calculate those from the data.
In about 6 weeks, we're going to have a talk from Elizabeth Wright, who runs the Cryo Electron Microscopy Center. What, if any, interactions are there between the kind of fluorescence microscopy that you do and cryo-EM?
Yeah, they're really complementary techniques. You're working with shooting electrons at your sample instead of light, and that's a major difference. But cryo-electron microscopy really gets you at structure. So you can see what does the individual particle look like, And from that structural information, it can really inform single molecule experiments. So whereas cryo electron microscopy, you get a static image. This is what the spliceosome looked like at this moment in time, at this part of the chemical reaction. We can take that information and we can be like, okay, what does the transition look like? What happens in between this image that we took with cryo-EM and this image that we took with cryo-EM, and actually look at the dynamics of, well, this protein left and maybe this protein came. And we can measure those kinds of things and how long this process takes, at least at the in vitro level. It's much faster in cells, but they're really complementary techniques.
Other questions? Here we go.
Other than tagging, how are your samples prepared? Are they on slides? Are they in tubes?
Yeah, so we have slides. We have to clean them really well, and then we passivate them so they have a certain coating, polyethylene glycol on them. And some of the polyethylene glycol molecules have a biotin attached to them, which we're able to use streptavidin to fix those RNA molecules to the slide surface. So we're doing everything on a glass slide. And then for our protein components, we have those in whole cell extract, and so we need a way to specifically attach a fluorophore onto our protein that we're interested in looking at. We use something called a snap tag. So it reacts with a specific substrate, which we can have attached to a fluorophore, and then you can do that specific reaction in whole cell extract and try to remove as much of that excess fluorescent reagent as you can. And that is what will flow onto the slide surface, which will interact with the RNA, is that fluorescently labeled protein in the whole cell extract. Did you have another specific question or follow-up to that? Or? Okay, yeah. Yeah.
One of the most amazing things I've ever seen is the bacterial flagellum motor.
Mm-hmm. Yeah.
Have any of you or any of your colleagues in the field made a cool picture yet of that
I
in
think
vivo?
So. You mentioned Liz Wright. I know there's some really cool cryo-EM structures of exactly what that motor looks like attached to the cell. And I think that maybe like super resolution microscopy, which is another single molecule technique, but it looks a little different from the movies that I've shown today. You're probably able to see it either like rotating, maybe with particle tracking, or with super resolution microscopy, it's more like fixed, and you can see different like color components as a part of that. And yeah, it's pretty cool.
It would be great to see that. I've seen cartoons of it, but to actually see real stuff,
yeah,
like whoa.
It's so neat.
I was wondering if you could say something, if you're familiar with it, about a process called MASER, which is molecular amplification by stimulated emission of radiation. I first heard of it back in the 60s and 70s, but haven't heard it mentioned much since. I was wondering if you knew anything about it. Thanks.
Yeah, I actually don't. I would be interested to know more about it now that you mentioned it, but I don't know.
You're shocked that you don't know something from the 60s.
Yeah, I do wonder if it's more like a materials like science technique, which single molecule is also applicable to that, and able to look at structures in materials.
But the big question is, what about phasers?
From Star Trek?
Yeah, yeah.
I mean, analogous to a laser, and you all know how that works, right?
What about quasars?
Quasars.
No, that's good. My goodness, it's 8:02. Must be time to.
Out of my wheelhouse.
Other questions? If not, thank you very much. It's fantastic.
Thank you so much.